Hi, I'm Monica Sentmanat, and today I'm going to talk to you about unbiased whole genome profiling of RAAV donor integrations with LOCK-seq. I want to start with a bird's eye view of what our core does, and that is to develop both in vitro and in vivo models to support therapeutic target discovery. For in vitro models, we reprogram cells from patient tissue samples to create patient-derived stem cells. For in vivo models, we engineer disease-relevant mutations in mice. Ultimately, whether you're working in vitro or in vivo, the accuracy of your model is only as good as your genotyping method. For today's talk, what I'm going to focus on is specifically mouse model creation, and I'll also focus on LOCK-seq, which is a long-read method to accurately assess multi-kilobase knock-ins. LOCK-seq stands for long-read sequencing of captured multi-kilobase targets. I'm going to go through LOCK-seq by example, first by starting with a comparison with another target capture method called Cas9-seq. Then I'll go through increasingly complex gene editing events and how LOCK-seq helps with genotyping these. I'll also go over probe and multiplexing costs and how to minimize those. And finally, bioinformatic pipelines for on and off target donor integrations, tools that can help with data wrangling and data interpretation. For nearly all gene editing projects, we use CRISPR-Cas9. The ability to knock in comes down to the efficiency of the double-stranded break and timing. So the availability of that donor template to be there at the right time and place. In the presence of donor template, a precise insertion or modification can be defined at that particular genomic location. In the absence of donor, an imprecise modification can happen, like a deletion or an insertion that's mediated through non-homologous end joining or microhomology mediated end joining. Ultimately, getting the reagents efficiently delivered into the cell is key. Gene editing in mouse embryos pose some unique challenges. Unlike cultured cells, the protective barrier around the mouse embryo makes it difficult for large molecules to pass through, and this is even when using an electrical pulse. Electroporation will not deliver circular plasmid DNA, linearized double-stranded DNA, or even very long single-stranded DNA into mouse embryos. And so for this reason, microinjection is required, and this is very labor intensive because it has to be done one embryo at a time. This can lead to low embryo survival due to the mechanical stress associated with using microinjection. It can also lead to random integration of double-stranded DNA, toxicity, and it also has very low knock-in efficiency. Unlike cultured cells, where you can target hundreds of thousands of cells in a single electroporation, here with every mouse session, so every day, you may only be targeting a few hundred embryos. And so ultimately, this can lead to several days of work and still only targeting 1,000 to 2,000 embryos, with only a fraction of those surviving the effort. One promising alternative to using plasmid DNA is recombinant adeno-associated virus. And this is a 4.7 kb single-stranded DNA virus with inverted terminal repeats. Each viral capsid is packaged with either sense or antisense strand, and it can infect dividing or non-dividing cells. And the DNA exists episomally, and in most cases does not integrate into the host genome. The two studies came out around the same time. One was Chen et al. In 2019 from UC Berkeley, and the other Yoon et al. In 2018 from the University of Massachusetts. And they both showed that you can efficiently transduce mouse embryos with a particular serotype of AAV, AAV6. And serotype really just refers to the selectivity of certain viral capsid for some tissue types. And both of these studies also showed that when you electroporate CRISPR-Cas9 in the presence of AAV6, you can install large knock-in events pretty efficiently, and this circumvents the need for microinjection. So what the workflow looks like is a pre-incubation step of the embryos with the AAV that allows for delivery of that single-stranded donor template into the embryos. And the next step is to combine the embryos with Cas9 guide RNA, RNP complexes for delivery via electroporation. And that then allows for that double-stranded break to happen, and with the donor template readily being available, should efficiently install that knock-in event. 3 to 4 weeks later, you should have F0s. So these animals that are born, they are chimeric animals in the sense that the gene editing event may have happened at different stages of development, of very early development. So it may have been at the 2-cell, 4-cell, 8-cell, and so on, whereby one cell may have gotten one flavor of gene edit and another cell at a different stage may have gotten a different flavor of gene edit. So these animals are genetically pretty complex. And in order to deconvolute what's happened, those F0s are first screened for the presence of the flavor of gene edit that you want. And those hits are then bred to wild type to identify germline transformants. In one example, we have generation of a fluorescent reporter, where we're putting in a 1.7 kb TD tomato knock-in at the end terminus of a gene of interest. And this donor template has 600 base pairs homology arms that flank the cut site. And this will facilitate homology-directed repair. The way that animals are typically screened is by using a 5' and a 3' junction PCR, where a primer is designed outside of the left homology arm that is genomic specific, and then one internal within the cassette for your 5' junction, and then the 3' junction one that is within the cassette or TD tomato, and then another genomic specific that is outside of the right homology arm. And the reason this is important to do is because it allows you to specifically look for a targeted integration event as opposed to a random insertion event if you were using internal only PCR primers. 67 animals were born from this effort, and 50 of them were positive for the 5' junction, and 48 of them were positive for the 3' junction. And in total, 47 were positive for both the 5' and 3' junction. Two things I want to point out is, although there's a very, very high success rate with 71%, there are still some animals that are positive for either just the 5' or 3' junction. And this can vary across projects, where some projects have a lot more of that discrepancy. And the second thing to point out is that it's not always clear in terms of the size of the band that it is the correct size. And in some cases, there can be variability in the amplicon size, which makes interpretation challenging. So with so many positive animals, how does one prioritize when you have so many to choose from? So we need a solution to better prioritize hits, because there are a lot of variables we want to be able to assess with a single assay. And these variables are not just looking at the correct knock-in at the on-target site by a gel image, but rather we are also wanting to know whether there are any point mutations or indels within that construct. We want to ensure there aren't any concatemers, that we don't have any random integrations elsewhere in the genome, or if there are, we'd love to know where they are so that we can follow them as we breed the animal. And we also need high enough sequencing depth to assay the full spectrum of events when you're trying to genotype a mosaic animal that has many events. And those events can happen at various frequencies depending on when in development the edit occurred. And the success of recombinant adeno-associated virus for donor template delivery, being that it's as efficient as it is, it allows us to multiplex. And so there's an even bigger bottleneck because there's a number of complex possibilities that can ensue. So you have multiple junctions that are required, and this can be very labor intensive. So having a different assay that allows you to do it all in a single shot is really necessary. Our solution to this is long read sequencing of captured multi-kilobase targets, or LOCK-seq, where we isolate genomic DNA from cell pellets or mouse tissue, and that genomic DNA then goes through either a mechanical fragmentation step using Kavaris G-tubes or enzymatic fragmentation using the cut and paste transposase TN5, whereby both fragmentation and the addition of barcodes and universal adapters are added to both ends of the fragmented material. For the genomic DNA that goes through Kavaris G-tube, the barcodes and adapters are added using just ligase. And so in the end, you end up with each sample being uniquely barcoded, which facilitates multiplexing in preparation for the hybridization capture step, where we incubate the samples with 5' biotinylated single-stranded DNA probes that are designed against the transgene or the specific region of interest. And we use streptavidin beads then to capture those fragments that bind to the probes and wash away unbound material. And that bound material then goes through a post-hybridization PCR step using primers that are specific to those universal adapters at the ends. And so there's an unbiased PCR step at this stage, and it's not primers that are designed against the specific transgene. Ultimately, these products are then going through the ONT library prep step, where we ligate the motor protein and load the libraries onto the MinION for sequencing. And so what I'm going to start with is just comparing LOCK-seq to another target capture method called Cas9-seq, and this is another method designed for the Oxford nanopore. And in this particular method, high molecular weight genomic DNA is first dephosphorylated, and Cas9 CRISPR RNPs are incubated for an in vitro reaction to cut the region of interest and thereby expose 5' phosphorylated ends that are now competent for ligation of the sequencing adapters. And so the only thing that has the motor proteins that are compatible to sequence on the flow cell should be those ends that were cut out of the genomic region of interest. So one thing I'll point out is up on the left-hand corner, I'll be distinguishing between the use of enzymatic versus mechanical fragmentation. In this particular example, for LOCK-seq, we use the enzymatic fragmentation with TN5, and we use SQL for that. So from mapped reads, what we noticed right off the bat is that compared to Cas9-seq, LOCK-seq did tend to have shorter read lengths. And this is, again, the enzymatic fragmentation method, where we see average read length for Cas9-seq being at about 3.8 kb and 1.4 kb for LOCK-seq. However, we noticed two really important other important factors. One is that the capture efficiency is starkly different. For Cas9-seq, it was at 1%, whereas it's 11.8% for LOCK-seq. And the number of mapped reads was much higher for LOCK-seq, and it allowed us to multiplex several more samples as compared to Cas9-seq. Looking at the IGV screenshots, we were looking at this region here where we expected a 432 base pair deletion, and we're definitely seeing reads with Cas9-seq that are consistent with our expectations. However, again, the depth of coverage was quite low, and this is not multiplexing samples. This is only running a single sample on a flow cell. While for LOCK-seq, we were able to multiplex 6 samples, and just looking at a single sample, we're seeing the depth of coverage is much higher, and we're definitely seeing the expected 432 base pair deletion. So in terms of performance, although the average read length was lower, the capture efficiency and coverage was substantially higher for LOCK-seq. And so what I've shown you is 10% capture efficiency and very high coverage for multiplexing 6 samples. And so this kind of multiplexability is really necessary for a screening tool, which is what we want to use LOCK-seq for. What I'll show you next is how LOCK-seq performs for a more complex edit. So to knock in even larger cassettes, we can co-transfect with more than one AAV to exceed the packaging limit of a single donor. And so this is a sequential targeting strategy where there's not just one guide RNA cut site, but there is a second guide RNA cut that happens after the first donor integration. And this facilitates the second donor integration event that now has 2 AAV sequential knock-in events in a single reaction. And so for this next example, we're going to be profiling this 3.5 kb knock-in with 800 base pair homology arms. For this example, we used the Kavaris G-tube for fragmentation, and we multiplexed 12 samples. Looking at the mapped reads, we found that the average read length was 4 to 5 kb, and our average capture efficiency was about 10% across samples. We also looked at the FASTQ files and looked for reads that were long contiguous reads that went across the entire cassette into the homology arms and across the flanking genomic DNA. And we were looking for reads that essentially supported targeted knock-in, and across several of the samples, we found many reads that indeed supported that. In the bottom 2 panels, we look at average coverage of the flanking genomic region versus the average coverage across the insert. And what you see is that for the average coverage of flanking genomic region, we are seeing high read depth, suggesting that we're able to target and capture the region of interest quite efficiently. However, not all animals have that insert. And in fact, the animals that are prioritized are consistent with those that have these long contiguous reads that support targeted knock-in. For at least one of the animals that we identified as a knock-in, we show that there's actually a concatenated knock-in where both AAV donors inserted downstream of the desired dual AAV knock-in. And between these events, there is an inverted terminal repeat. Just one note is that clearly 5' 3' junction PCRs would not discriminate an animal that has a concatenated knock-in event versus one that has a single insertion event. In this example, we use the Kavaris G-tube to fragment the DNA. And again, we see 4 to 5 kb read lengths for the mapped reads across the 4 animals we multiplexed in this particular run. Among the animals we multiplexed, one was M24, which is the founder F0 animal, and that animal was then bred to wild type and produced the 3 progeny, F19, F21, and F20. Across all 4 of the animals, we were able to identify these long contiguous reads that map across the entire cassette and flanking genomic. However, when you look at the per base coverage across all the animals, we observe some deviations from expected read depth within the insert, and these elevations in read depth suggest that there could be a gain in copy number for these regions. Digging into the data, we identified two random integration events. Both were partial AAV genomes located in coding regions, and they were both present in the founder M24 animal and in 2 of the 3 progeny. What I've shown you is that you can use mechanical fragmentation for even longer read lengths, and that there's sufficient coverage to screen founder animals for both on-target and random integration events. Next, we'll take a look at 3 AAV knock-ins. In this example, we have a 3 AAV knock-in where we have 2 AAVs that are constant, donor 1 and donor 2, and there's a third AAV that's a variable AAV donor where it has one of 3 different flavors of knock-ins, a wild type, a 54 base pair deletion, or a 432 base pair deletion. In the end, we generated 3 transgenic animal models, one with the full-length wild type and 2 with one of the 2 mutations. And the total insert size was 6.6 kb with 800 base pair homology arms. We used TN5 for fragmentation in this example and multiplexed 6 samples. Across the samples, our read lengths of mapped reads were around 1 to 2 kb. And we were able to identify across the 3 models the expected mutations that we installed, either the 432 base pair mutation or deletion, which has this reduction in read depth for the model that is 432 base pair deleted. We have the 54 base pair deletion, and the wild type has those regions present. However, in the wild type sample, we did see a deviation from the expected coverage. That deviation was found to be a concatenated insertion event where one of the AAVs is integrated downstream of the right homology arm. And this was present in just the wild type animals and not any of the others. This was also confirmed by PCR and Sanger. There's several examples of unrelated projects that have imprecise insertion events that could give false positive junctions. In this first example, we have an imprecise knock-in at the 5' end, where there is the insertion of part of a right homology arm downstream of the left homology arm, as well as a 132 base pair insertion in the middle of the CAG promoter. In the bottom, we have an internal deletion of one of 3 SV40 inserts, and that insertion event is present in an otherwise correctly targeted knock-in. In a different example, for a much larger insertion event, there's evidence of a missing insulator and an upstream enhancer region that's absent from all the animals. And what this does is it really gives you a lot of clarity over what the configuration is at the site compared to junction PCRs, where only one and not the other junction is present. And this opens the door for easier troubleshooting and possibly retargeting partially targeted animals. I've shown you that in spite of the shorter read lengths we get with TN5, they're still long enough to call imprecise knock-in events. Next, we're going to go over a couple of hacks to overcome some of the costs associated with probes and multiplexing for LOCK-seq. Probes tend to be 120 mer biotinylated single-stranded DNA. They're typically 5' biotinylated and are spaced either at high or low density. And when high density, tiling occurs, it can be partially overlapped to adjacent probes to ensure full sequence is covered evenly. And there are custom designs that can be available from any commercial supplier like IDT, Twist, and Agilent. For each project, you are likely to require a unique set of probes. For a commercial designer, the easiest thing to do is to send the region of interest to the designer, and they will generate high density tiling panels for that region. And the caveat is that although the cost per probe is relatively low, there's a limited number of reactions that you can use that panel for. Alternatively, you can design in-house designs and have low tiling density and have a limited number of probes. The advantage of this is having a much higher number of reactions available to you. However, the upfront cost is high. To reduce cost and give us a lot more flexibility in probe design, we used an in-house probe protocol for LOCK-seq using asymmetric PCR, where we first perform a first-step PCR to generate amplicons for the region of interest using tailed primers. And then in a second step, we do asymmetric PCR with the first step amplicon as input. And in this reaction, we use a very low amount of reverse primer with forward biotinylated primers. And then all of the amplicons are pooled and cleaned up, which could then be used directly for a hybridization reaction with no further downstream work. This in-house probe protocol really gives us the lowest cost per hyb and the lowest cost per probe. It again gives us the versatility to design varying tiling densities and probe lengths. In this example, we targeted a 1.3 kb region and used DNA fragmented with Cavaris. We were able to pull down comparable read lengths as we had with other probe sets that were commercially supplied. 3.3 to 4 kb read lengths. Our target capture efficiency was 10 to 20%, and on-target mapped reads from 3 to 17,000. So these numbers are all very comparable with commercially supplied probe sets. Digging deeper into that data, we were able to identify these raw contiguous reads that overlap flanking genomic regions. So this suggests that we're not missing out on these on-target long reads using these probes. We were able to successfully assemble consensus sequences, and in some of the animals, we were even able to detect concatemers, which again suggests these in-house probes perform as they need to. Switching gears to multiplexing, TN5 is a lot more amenable to high throughput because Cavaris G tubes aren't supplied in 96-well format, and so it is not as amenable to high throughput as TN5 is when you may have dozens of samples that are required for tagmentation or fragmentation and barcoding. The per sample cost can be high, however, and so if this is performed a lot in a lab, using TN5 from an academic source or made in-house can circumvent that cost. One of the caveats is that it requires a bit more upfront work because with each lot that is generated, it requires preloading the TN5 with the oligos containing your barcodes and adapters. And it also requires testing that lot for activity so that you use the right amount of protein and in the right conditions to get the longest fragments. So that upfront work can really pay off if this is being used for high throughput screening on a regular basis. In this example, we used tagmentation using TN5 from an academic source, and with crude lysate from 59 iPSC clones, we screened and identified many that are positive for knock-in. We prioritized the clones based on mean read depth across each feature of the transgene and found that the top 4 clones identified by LOCK-seq were also positive for the 5' and 3' junction PCR. The cost per clone is only a couple of dollars for this method, and it supplies you with sequence data so that you can then verify the integrity of that knock-in event in addition to prioritizing clones. Shown you that in-house generated probes perform comparably to commercially supplied ones, and they give you a little bit more versatility with the ability to play around with probe design and tiling density. I've also shown you that non-commercially supplied TN5 can be adapted for high throughput screening for long read sequencing platforms. Although it requires a little bit more upfront work, it can really save costs if it's something you plan to do routinely. Next, I'm going to talk about data wrangling and data analysis and some tools that we routinely use for analyzing our ONT data. So once you have your data, there are a couple of scorecards you want to fill out. The first relates to quality control, and it has to do with ensuring that the capture and that the sequencing run performed as expected, and that they're adequate enough for you to have faithful calls in the full-length knock-in event, but also that the data is able to report the presence of concatemers, partial knock-ins, mutations, and random integration events. And if you do find things like this, that they're not necessarily a product of artifacts, but rather true to the genome that you're sequencing. In-house, for the first step, what we typically do is just map the FASTQ files to the transgene, or in other words, just the knock-in event. We're not necessarily starting by mapping to the genome. And once we have those mapped reads, they're BAM files, and we effectively just use SAM tools to output the alignment statistics. That's your flagstat file, the per base read depth, using the depth flag for SAM tools. And we also grab the mapped reads from the BAM files. And that's our FASTQ file that we use to assess read length, mapped read length. The flagstat output looks like this. And what this will give you is on the top line, the total number of reads for that sample. And on the 5th line, the total of reads that map to the reference. And again, in our case, where our reference is just the transgene. And so here we have the total mapped reads, and then we're using target capture efficiency as 26% in this example, as these are the mapped reads that of the total reads that were generated from this sample. For read lengths, we are using the mapped FASTQ, and we're using a very simple command line that essentially just analyzes text-based data. And we're grabbing the DNA sequence from each identifier and counting the number of bases that are present along each of the mapped reads so that we have an output file that tells us the sequence length for each of the mapped reads, and that is what we use to plot for each one of the samples to give us a distribution of the length as we change protocols, try different methods to enrich for larger and larger read lengths. This is what the depth file looks like. And in our case, again, we're using the transgene. It was just called untitled, so that's what the chromosome name is. The second column is the position along the file or sequence, and the third column is the depth of coverage for each one of those positions. This information can help you identify low depth or missing regions, compare coverage between samples, detect potential CNVs like we saw in previous examples, and filter out these unreliable positions maybe for variant calling. Such unreliable positions tend to be homopolymers. This is particularly true for ONT data, whereas your homopolymer length increases, you have less fidelity in sequence quality, and so you tend to have slight underestimation of the length of that homopolymer where you may miss a nucleotide. In the next step, we take these mapped FASTQ files and we use a tool that's CANU, and this tool helps build consensus sequences for noisy reads, and it performs error correction, trimming, and can handle repeats, and you can adjust the minimum overlap length for assembly. It outputs a FASTA file, and it will assemble contigs associated with these mapped reads. The CANU tool will really help confirm precise on-target insertion by assembling the mapped reads, and it will output however many contigs it can assemble from those reads. You supply the genome size, and since in the first pass the reads were mapped just to the region of interest, it would be pretty small, so maybe 15 kb. The max input coverage allows you to downsample the total number, the amount of data, and so the runtime, it can prevent excessive computation time and increase the speed. The min overlap length is essentially the required overlap length between two reads for them to be assembled, and the default for this is 500, but you can increase this if you have, for example, like a very repetitive region. And the core out coverage uses up to 100x coverage for error correction, and the default is 40 to 50x. Core min coverage is the number of reads required, essentially for a contig to be called or assembled. And essentially, this is an example of what the output file looks like. This is the file that gives you the contigs, and you can essentially just open this file in Snapgene and assemble all the contigs onto your transgene, so that would then verify the on target, and then it also gives you a sense of whether there are any coding variants identified along the length. So there's a couple of ways of verifying the full-length insertion. We saw examples of using the raw full-length reads that went across the entire transgene, and you're able to do that if the transgene is small enough, where the read lengths exceed the size of the insertion event. However, this isn't really doable if the knock-in event is so much larger that it exceeds the average read length for your run. And so in these cases where you have a very large insertion event, assembly of the contigs is going to be your best bet. So whether if you have a very small transgene, you are able to use these single raw reads for full-length coverage. It's very easy to even detect concatemers, and 5' and 3' genomic with transgenes in a single read is something that's quite achievable. However, when you have very large transgenes, you will not find these single reads that will traverse across the entire region. And you will be able to detect concatemers, but again, using a tool like CANU is really essential to assemble these events for large insertions. To get at random integration events, those mapped FASTQ files can then be run through, again, Minimap and SAM tools. And in this case, when you're using Minimap, you're using your genome as your reference. In our case, we have mouse samples, we're using MM39. And the output from SAM tools is our depth file. And you can also throw in the map FASTQ files here. In this case, your depth file will now have the chromosome location, the position along that chromosome, and the depth. And so this is now just not untitled, but rather the genomic coordinates. And in this example, we have two random integration events, one on target, and this particular event failed to confirm by PCR. But as you can see, the depth of sequencing is very, very low, and so this is likely some kind of background. In order to assess the configuration of these random integration events, you can extract these regions from the BAM files and use some window, 2 KB window on either side, for example, convert those extracted reads to FASTQ and run CANU on them. And using this output, you're able to very easily assemble what the configuration is at each one of the off-target sites, and then follow up with some confirmation like Sanger sequencing, just to verify that this isn't an artifact. And in our experience, they're not typically artifacts when you have very high read depth, especially if it's similar read depth to the on-target site. So I've shown you how we use Minimap, SAM tools, and CANU for assessing on and off-target integration events. And these are things that we use to first pass prioritize animals and assess quality of the data. I'd like to thank the folks that worked on this project. They include our core director, Xiaojia Cui, the folks that lead the molecular biology work, who include Jane Kornova. Technical execution of LOCK-seq is Wang Ching Chen. We also have bioinformatics support from Jed, Sam, and Noah. And in vitro validation of all the reagents used for the mouse projects was performed by Zi Wang. We work with 2 mouse cores here on campus, the transgenic knockout and microinjection core and the mouse genetics core. They were responsible for making the animal models we screened. AAV vectors were generated by the Hope Viral Center. I should say the production of the virus was made by them. Sequencing of some of our samples was performed by the Center for Genome Sciences and System Biology Sequencing Core. And I'd like to also thank Dave Briner and Dan Hughes for help with the presentation, and they're here at the McDonald Genome Institute.